Okay, so here we are in chapter 25 talking about metabolism. This comes right after chapter 24 on the digestive system on purpose. So we're going to see a lot of connections here. Now, I'm going to skip over chapter 25.1 really is a review of your nutrition class. I pointed that out in the module. Do go ahead, do the smart book for this. Use that as a review. If you notice that you're deficient, you've forgotten certain areas of that, you can go back and study that. But I'm not going to go over it. I want us to jump right in to section 25.2 and think about metabolism. So metabolism is the sum of all the chemical reactions that take place in the body. And typically we are going to divide that into two categories, catabolic reactions and anabolic reactions. Now, catabolic reactions take complex molecules, say a whole protein, right? And they break them into smaller molecules, say individual amino acids. Or what they've shown here is something like maybe taking a carbohydrate, running it through all of cellular respiration, which we'll talk about, and ending up with really simple molecules like CO2 and water, right? So catabolic reactions, picture a cat, right? Tearing things apart, ripping molecules apart. You go from complex to simple. Now, in the process of that, you release energy. Catabolic reactions are exergonic. They release energy. And so that energy can be released in the form of heat. We try to capture a lot of that energy in a molecule called ATP. And that's going to be the cellular currency of energy, the type of energy that cells know how to use. So our other group of reactions then are anabolic reactions. These are reactions that build more complex molecules, right? Think anabolic steroids, right? And trying to be like a muscle person weightlifting, right? You would take amino acids and put them together into these more complex forms of proteins, right? So that's an anabolic reaction, going from simple to complex. Anabolic reactions are endergonic, they require energy. And so ATP serves as this nice link between catabolic reactions and anabolic reactions. And so if you think about your diet, right, you're eating these different macromolecules, and they really do two things for you. They can be broken down for energy, which we'll focus on a lot in this chapter, but they also then provide, right, the end of those catabolic reactions are actually the simple building blocks, the individual amino acids, the monosaccharides, that we can then use, as long as we have energy, to make things that are more complex, right? To make cell membranes, to make antibodies, etc. Okay, so that, when we talk about metabolism, this is what we mean. All of these different chemical reactions. Okay, so this is an image. Sorry, I was trying to make the screen bigger. 25.3 is kind of reminding us about ATP. So remember, adenosine triphosphate. This is the molecule that's required for cells to do a lot of work. And so what ends up happening here, and I like that they did this, right? They're showing us this process of phosphorylation, right? So if we take ADP and phosphate and we put them together, it takes energy, right? So that energy that comes from these catabolic reactions, I kind of hate the way they labeled this, but the energy coming from the breakdown of your foodstuffs is able to push these two molecules together. They're actually both negatively charged. They don't want to go together. We force them together, and then the energy stays in this bond, right? So ATP is really a form of potential energy. And when we decide we want to do an anabolic reaction that requires energy, then we'll go ahead and break that apart again into ADP and phosphate. The release of energy is utilized by anabolic reactions. Okay. Another way to think about this on the cellular level, right, is one of our big goals, and what we'll see in section 25.3 is really about making ATP, because ATP is how cells do all these things, right? Run transport, locomotion, exocytosis, you name it, right? And then also gives us that energy to run anabolic reactions, right? To build more complex proteins, right? So that we can do things like grow and repair and secrete, etc. So what they're showing you here is that the end result of digestion, essentially catabolic reactions, is that nutrient pool. We can use those individual amino acids to build more complex molecules. And then a lot of that nutrient pool also, right, notice the size of the arrow here, a lot of that nutrient pool goes into aerobic metabolism in order to produce the ATP that we need. So we really have a couple of different options here. Okay, so what we're going to focus on as we look at cellular respiration is we're really going to follow glucose. But what I do want to point out is that we do and we can use other fuel sources. So I really like this, right? Think of our nutrient pool, all the foods that we ate and broke down. Fatty acids could be stored as triglycerides. Glucose can be stored as glycogen. Amino acids can be built into proteins. But what we also see is that all three of these, the amino acids to a lesser extent, but all three of these can get put through aerobic cellular respiration to make ATP. And I do think that's important for us to realize that all three of our macromolecules can be utilized as fuel sources. Okay, so big picture, because we're about to get into the nitty-gritty, right? But the big picture here, what we're looking at in aerobic cellular respiration, that's where we're going, 25.3, is this idea that we can turn our food into ATP, a form of energy that our cells know how to use. Here we see a cell using ATP in order to run the sodium-potassium pump. Here we see a motor protein dragging an organelle right along the cytoskeleton of a cell. Here we see energy, this is tiny, energy being used to put glucose together to make glycogen. Energy being used to put fatty acids with a glycerol and make triglyceride. Energy being used to put amino acids together in proteins, right? We have a huge need for ATP. So here we go. We are going to jump in and see how we get that energy. So aerobic cellular respiration. What we're going to do here is we're starting out with a glucose molecule. So we are going to follow glucose. That's pretty standard, even though we just saw those other macromolecules can get used as well. So the first step in aerobic cellular respiration is glycolysis. Literally, if you look at the word, lysis is like a splitting. We are going to split this glucose. Glucose is a 6-carbon molecule. I'm going to write that, right? Glucose is a 6-carbon molecule. And we are going to split it in half into 2 pyruvates. Each pyruvate is a 3-carbon molecule, right? So we haven't lost any carbon yet, right? But what we have done, and it's a more complicated process, we're going to keep this fairly simple, but in glycolysis, in that multiple metabolic steps that get us to pyruvate, we are going to end up producing 2 ATP. We actually produce 4. Do you remember this, right? You have to put 2 in to kickstart things, you make 4. So at the end, we end up net result getting 2 ATP. We also get 2 of this molecule called NADH. Now, NADH, what we're dealing with is we have this molecule called NAD, nicotinamide adenine dinucleotide. NAD, right? NAD+. This is an electron carrier. So what we're going to see over and over again here, right, is that this molecule, NAD, is willing to be reduced to this form NADH. Do you guys remember that terminology, reduced, right? Anytime you gain an electron, that's considered reduction. Lose an electron is oxidation. These are good things to remember. And so this is the reduced form when we're carrying, biologists, when chemists talk about oxidation and reduction, they're talking about electrons. And for us in biology, that electron is going to be on a hydrogen. So you can follow the hydrogen here. So this is the reduced form. It's carrying that electron because it has a hydrogen. And this is what we would call the oxidized form. And so what we're going to see is that this reaction can go either way. We are going to spend a lot of time here towards the beginning of respiration, gathering electrons and taking them, notice over here, right? Taking them to the electron transport chain. So anytime you see this NADH, it's just carrying electrons to the electron transport chain. This is kind of an intermediate form of energy, right? I should also point out, remember with these oxidation reduction reactions is that anytime they go together, anytime something is reduced, something else had to be oxidized. Anytime something is oxidized, something else has to be reduced. And so in this case, when we look at aerobic respiration, we are trying to oxidize glucose, completely rip all the hydrogens off of that. And you can kind of see it if you look at the overall equation, right? You're taking glucose, C6H12O6, and you're splitting it into carbon dioxide and then water. You've taken all the hydrogens off of the glucose, right? So you end up with CO2. There's no hydrogens on there. We've moved them around. Anyway, so this is going to the electron transport chain. Okay, so in glycolysis, we split glucose into pyruvate. We make 2 ATP and 2 NADH. All right, so those 2 pyruvates, then we've got options. If there is no oxygen, and we can come back to this, right, we can run fermentation. That doesn't require oxygen. It's going to end in the production of lactic acid. Lactate and lactic acid are the same. But we only end up getting the 2 ATP here from glycolysis. So it's not it's an incomplete oxidation of glucose. It's not particularly efficient. We don't get a lot of ATP there. So what we'd like to do is where we're heading here is aerobic respiration, right? So if there's oxygen, we're going to take this into the mitochondria and run with it, okay? Okay. So, wow, there's a lot going on there. Maybe we'll come back here and then we'll dive in if we have to. Let's use this one for now. The second step, I wish I could type on here. I can't type on here. So really, glycolysis is this first step, right? There's a step going on right here, that's a 2, called pyruvate oxidation. And basically what we're doing in this case is we are taking pyruvate, we're converting it to a molecule called acetyl-CoA, which is going to feed into the citric acid cycle, okay? In this pyruvate oxidation, sometimes called the linking step, we're actually going from 2 3-carbon molecules to 2 2-carbon molecules. And so the carbon that we're losing is in the form of CO2. So during the second step, pyruvate oxidation, we actually start exhaling, right, some of this CO2, and we make 2 more NADH. So notice we didn't make any ATP at this step, but that NADH going to the electron transport chain is going to contribute to the overall ATP production. Okay, so then our third step is inside of the mitochondrion, and that is the citric acid cycle. Right, and we'll look at this in a little more depth, but you do not need to know all 8 intermediate steps, the enzymes involved, etc. Okay, what you do need to know is that in this process, essentially what's happening, we're taking this 2-carbon molecule that we just made. It's called acetyl-CoA. It comes into the citric acid cycle, and it actually, so we had a 2-carbon acetyl-CoA. It bonds with a 4-carbon molecule called oxaloacetate to make a 6-carbon citric acid, and then that molecule is going to run through the cycle, and we're going to rip those carbons off, right? Don't know where there we go. And we come back with the oxaloacetate to grab the next acetyl-CoA. It cycles through, right? And that goes on and on. That's why it's called the citric acid cycle. And so for each glucose molecule, right, this needs to run 2 times. And that's actually the totals that they're showing us here. So each rotation of this is actually making 3 NADH for a total of 6 NADH per glucose. We make 1 FADH, but it goes around twice, so we get 2. We make 1 ATP, twice equals 2, and we make 2 CO2, which goes, again, 2 for each acetyl-CoA. Oh, look at that. Acetyl-CoA is a 2-carbon molecule. If you make it into 2 CO2, this is actually when we complete the oxidation, the removal of electrons from our glucose, right? And so here's the rest of our CO2 waste product. We're going to be breathing that out. Oxaloacetate comes back around, grabs the next one. Okay. So here we see actually quite a bit of NADH and FADH. Sorry, FADH is flavin adenine dinucleotide, just another electron carrier. Notice, so nicotinamide adenine dinucleotide, this is based off of niacin. FADH is based off of riboflavin, but they're both electron carriers, and this is going to the electron transport chain. So at this point, we still haven't made much ATP. We've only got, 2 ATP from glycolysis and 2 ATP from citric acid cycle. What we're going to see is that our electron transport chain is going to do the majority of the ATP production, and we'll take a look at that. Okay, so this is a nice image, other than the ugly brown color. This is a nice image because it shows us these steps, right? So here's our one. So we're going to look at this a few different ways. Here's our glycolysis, right? We took the 6-carbon glucose and we make 2 3-carbon pyruvates. Very nice. They're showing us the 2 ATP and the 2 NADH. They're also, what I do like is they're showing this takes place out in the cytosol, right? It doesn't require any oxygen. And then as we start to move into, as we go on in our steps, we're moving into the mitochondria. Okay, so our second step here is that pyruvate oxidation. So taking our 2 3-carbon pyruvate molecules and making 2 2-carbon acetyl-CoAs. We exhale 2 CO2, we send 2 more NADH to the electron transport chain. Okay, our third step then is the citric acid cycle. 2 acetyl-CoAs come in, join this 4-carbon molecule, which is called oxaloacetate, and we make citric acid. And again, they're showing the results of 2 turns of the wheel, right? So if glucose got split into 2 pyruvate, and then each pyruvate got converted into acetyl-CoA, one goes through, and then the next. Okay? So we end up with 4 CO2, 6 NADH, 2 FADH, 2 ATP. Yep, and then we're back to oxaloacetate. Great. The fourth and final step, we're going to zoom in and look at this. The electron transport chain in chemiosmosis is going to take place across this inner mitochondrial membrane. So they're really trying to emphasize here, notice where we are in the mitochondria. We've got all these like little internal folds, the cristae, and this space outside of there. And that's really where this reaction is going to take place, this inner mitochondrial membrane. Okay, again, this is what the citric acid cycle really looks like. And this is where I was like, you don't need to know all of these intermediate steps. If you take biochem, you'll get that for sure. Okay, so here is this fourth and final step. Now, what I typically call this step is oxidative phosphorylation. Okay, so what we're going to see is oxidation taking place, right? So some molecule has to lose a bunch of electrons, and that is going to drive phosphorylation, the addition of a phosphate onto ADP to make ATP. All right, so here is how this fourth and final step, oxidative phosphorylation, takes place. A big part of it, it's kind of like 2 steps here. The electron transport chain. This is the part everybody always remembers. Like, okay, there's all these proteins embedded in the inner mitochondrial membrane. This is what's happening. All the NADH and FADH, right, they've been running around carrying electrons, they get oxidized. So notice here that we go back to our oxidized form, NAD+, or FAD+. The hydrogen has electrons, an electron and a proton. So notice what happens here. The protons are getting pulled into this inner mitochondrial, the space between the 2 membranes, the inner membrane space. The electrons are getting pulled into this electron transport chain, and they are going to keep getting handed down through the electron transport chain, right? They keep moving to like a slightly lower energy level. Eventually they'll come back out the bottom inside the mitochondria. One thing, I don't usually emphasize it too much, but while we're here, let's take a look. So one of the things that happens as we're passing all these electrons, it actually allows some of these cytochromes, some of these proteins in the electron transport chain, to actually pull hydrogen molecules against their concentration gradient, basically to pump them into this inner membrane space. Heck, look at this. Look how many hydrogens are ending up outside of this inner membrane, right? So that's the electron transport chain. Now, what we're set up for is what's called chemiosmosis. So remember, molecules don't like to be like all clustered in one side. They want to diffuse, they want to spread out. And so that's exactly what's going to happen here. All these hydrogens, they're really crowded in this inner membrane space, but there's only one place that they can cross back into the matrix of the mitochondria, and that is in this protein called ATP synthase. So notice what happens. The hydrogens fall back through here. And literally the ATP synthase is like a turbine. Each hydrogen starts kind of ratcheting it around. And think of this membrane as a dam. There's a big reservoir of water behind it, and then we put a hydroelectric turbine in. It's the same thing, but now we have hydrogen, we have protons, right, that are going to flow through here, turn the turbine, and instead of making electricity, we harness that energy for phosphorylation, right? So to make ATP. So cool. Okay. And you're going, there's way too many details. This can't possibly be cool. When you get the hang of this, and hopefully between Gen Bio and here and micro, like we're starting to grasp this, right? So notice those hydrogens, the protons have come back through, the electrons have come back through. This is where we finally use oxygen. Oxygen is going to be reduced, right? Oxygen is going to gain those electrons and become water. Oxygen is the final electron acceptor in cellular respiration, and we produce water. So cool. Okay. So that is a very kind of quick run through this, but that is the level that I feel like you need to understand this. Tons of great videos out there. Khan Academy has great resources on this. My guess, I haven't looked, my guess is Crash Course does as well. So spend some time if this didn't really sink in Gen Bio, we can certainly go over this more in class. But I kind of wanted to make sure that you understand the level that I want you to know that at. Okay, so that is, 25.3 is really just following the metabolism of glucose that way. What we're going to look at, honestly, much more briefly, is the way that we use other macromolecules. And so this is looking at how we can use lipids, right? So there's this process called beta oxidation, where we can actually take fatty acids, right? So remember, a triglyceride is a glycerol and 3 fatty acid tails. You can store so much energy in a lipid, and what we're going to do is we're going to rip it apart 2 carbons at a time, basically in this form of acetyl-CoA. And so our lipid gets fed into respiration as acetyl-CoA, right? And so beta oxidation takes those fatty acids, makes acetyl-CoA, and then we can run through this and get ATP. Okay. One other thing I would point out, right, is that the metabolism of lipids can lead to what we call ketone bodies. And this will become important in diseases like diabetes, where the cell can't use glucose, right? Insulin's not letting the glucose in. And so we switch our metabolism and try to get fats. But honestly, for the most part, cells are super stoked to use triglycerides, right? So glucose tends to be reserved for like periods of intense activity. Most of the time, a lot of your cells are just working with the stored triglycerides, the fatty acids that we have. Okay, so then this picture is looking at, remember you have 20 different amino acids. What they're showing you here, and you don't have to memorize this, but what I do want you to realize is that amino acids actually get plugged into cellular respiration in lots of different places. And it really just depends on what the structure of that amino acid is, right? So some end up looking to the body, once you rip the nitrogen group off, once you do deamination, it looks like pyruvate, and you go from there. And others end up being 2 carbon molecules that look like acetyl-CoA, and so we go from there. And then there's a few that kind of plug in at various intermediate steps in the citric acid cycle. So just know that amino acids can get used, and they're a little different. One of the things, so there you go. I'll come back to that. One of the things that maybe is important is realizing, once you go to acetyl-CoA, you can't go back to glucose. We don't have the enzyme that would allow us to turn acetyl-CoA into pyruvate and therefore go back to glucose. From pyruvate, you can. So when the liver is doing gluconeogenesis, right, it's actually using these amino acids because those are the ones that can convert back to glucose. Anyway, food for thought. Okay, so I did want to point out, right, as the body starts using non-glucose sources, particularly those lipids, what we can end up with are what are called keto acids. Again, we see this as a result of lipid metabolism. They're trying to point out here that there's actually a lot of similarities to an amino acid. For us, probably one of the most important things to know is that it is, in fact, an acid, and so it can donate a hydrogen. And so one of the things you'll see, particularly in diabetes, is if someone's doing a lot of this metabolism, they can often become acidotic. Okay, other thoughts in protein metabolism here. I mentioned deamination, right? And so what they're trying to show you here is that we have ways to basically remove, see how this nitrogen group is attached? Over here, it's ammonia, it's separate. And so deamination is the removal of this nitrogen group. But notice it actually changes our molecule. We no longer have glutamic acid, right? Because we've taken NH3 off. Now it's alpha-ketogluteric acid. You don't need to know the specifics, but you should know what deamination is. In transamination, this is how some of our amino acids are not essential, right? You don't have to consume them in your diet, because what we can do here, right, is we can actually move the nitrogen group from one molecule to another, right? So here we have an amino acid. Just put it in a little box here. And if we add this alpha-ketogluteric acid, which does not have a nitrogen group, notice we can switch where the amine group is, right? We can go back to having glutamic acid, and we can switch this one. Anyway, transamination is the transfer of amines between molecules. Deamination is the removal. One thing to note, right, is that as you're deaminating, as you're pulling nitrogens off and making ammonia, this is useful, right, because this now looks a lot like a sugar. We can use this for ATP, etc. But ammonia is actually really toxic. And so one of the things we do in our body is we convert it into a less toxic molecule called urea, which you see here. And so super high protein diets, as long as your kidneys are okay, you're probably fine, right? Your kidney can remove quite a bit of urea, but for individuals who have kidney illness, it's probably a bad idea to make them work extra hard, because it is the kidney who has to get rid of all this nitrogen waste, this nitrogenous waste. Okay, so in the PowerPoint that I'm leaving up on Canvas, it runs through with some different pictures, the same thing. Feel free to flip through those if you like them, study off of them, right? It's the same material. They're a little more colorful, but otherwise, your book actually did okay in my mind in this section. So I did leave those up. Let me just flip through there. Again, I pulled up a couple different ways that different books, this is actually out of the micro textbook, how they summarize the steps of aerobic cellular respiration. This one, again, kind of goes all the way through. I like that they even start with glycogen, right? And they say, oh, glycogenolysis can get me glucose. Glucose can go to pyruvate, right? Etc. Beta oxidation. We were talking about these lipids last week, so if you want, you can see how LDLs and HDLs are made, but we did cover that in chapter 24. Okay, so then the last thing I wanted to point out in this chapter is thinking about the differences between the absorptive and the post-absorptive state. So this is section 25.7, and basically what they're showing is like when you're eating and shortly after. I'm going to switch back to this picture. So here they're trying to show, okay, you ate breakfast, you're eating lunch, you're eating dinner. They're showing these like spikes in nutrient levels, glucose, lipids, and amino acids. And so, as you're eating, immediately after you're eating, we consider this the absorptive state, right? There's maybe a little delay till those nutrients are circulating, right? You ate breakfast and there's our absorptive state, this peak. And so what I want you to see, what they're trying to emphasize here is all of these anabolic reactions. So they're doing anabolic reactions in blue. So we can see, right, that we're making fatty acids, we're making triglycerides, right? We've got so much glucose in our blood that maybe we're making glycogen, right? Those amino acids that came into the pool, we're building proteins, right? We're definitely also producing some ATP here. And so we do see glucose being broken down and being converted to ATP. But there's this real emphasis on anabolic reactions and the storage of nutrients and energy. Again, this is your book, and they're saying the same thing. It's just all brown. The nutrients are absorbed, the nutrients are processed, and really everything is anabolic, or most of it is anabolic, right? We're making proteins, we're making glycogen, we're making adipose, we're etc. Okay, that's the same picture again. In the post-absorptive state, and so here they're showing overnight. This is a beautiful example, right? Maybe you had dinner at like 6. See how these nutrient levels are falling. So we hit this post-absorptive state overnight as we sleep. So now notice what happens. All these lines are red, which are indicating catabolic reactions, right? So we take lipids and we break them down into fatty acids and we feed metabolism, right? We're taking glucose and we're splitting it and we're making ATP. Our proteins are getting broken into amino acids, which are being fed into the cycle. So the emphasis is much more on these catabolic reactions because we don't have this constant input, right? Notice they are pointing out here when you're utilizing those fatty acids, we are making those ketone bodies, those keto acids. And so again, overnight, that's probably making a slight change in your pH, but within physiological boundaries. In someone with diabetes, those levels could be really high and push them into acidosis. Okay, again, same thing from your book. Again, just notice catabolic, right? We're tearing things apart here. We're trying to release those stored energies. Okay, and then the same picture. I don't know, I have this all twice. Okay, last thing in this chapter, and then we will get you out of here. I guess they just couldn't figure out anywhere else to put this. And so we're looking at a little bit about how the body regulates temperature. So this is section 25.9. And so pointing out the difference between things like radiation, right? The exchange of heat between things that actually aren't in contact, right? You're getting heat radiation from the sun. Conduction, right? She's touching the sand with her foot there. So the exchange of heat when things are in direct contact. Convection, this is the exchange of heat between things like our body and the air or the water. Okay. Evaporation, we use sweat, but even just say getting out of the ocean there, right? That water on the surface of your skin, it actually takes a lot of energy to make that water go from an aqueous form into a gaseous form. And so we're able to pull heat away from the body. Same picture just from the 70s, little jazzercise here. So one thing your book didn't go into, which I did want to take a quick moment for, if we're going to bother thinking about heat loss and heat gain, is thinking about how your blood vessels help you adapt to this. Let me get out of the way here. Right, so notice in a warm environment, right, we're doing things like opening up these blood vessels that are much more superficial, right? They're showing you ooh, remember we just learned the cephalic vein, that one that was out kind of on the deltoid. Very superficial blood vessels. These are going to be dilated. We're allowing more blood flow in that warm environment. We want that blood flow to go to the surface of the skin and be able to be dissipated. In a cold environment, we're going to go ahead and do some venoconstriction there and try to hold that heat in towards the core. I know it's hard to tell, right? But think about where that brachial artery is. It's really tucked in under that bicep, and so it's much easier for it to stay warm. I did want to point out we use this countercurrent exchange, and this, to me, countercurrent exchange is one of the coolest things in biology. We'll see this again in the kidney, but think about the way this works. So you keep running these blood vessels with each other the whole time, right? And so think about this. The brachial artery is going out towards the hand, but it lays right next to the brachial vein, which is going back to the core. And so one of the things that we do, and it's really just through diffusion, right? Energy moving from high to low. Hot blood heading towards your hand is actually warming the blood that's heading back to the trunk. Okay, and so any cooling that took place maybe out in that hand, we're really able to protect core body temperature, which, I mean, frostbite's a bummer, right? Maybe losing a finger would be sad, but it's better than actually moving into hypothermia, getting that core temperature to drop to the point where you die. So I did think that was pretty interesting to think about how our body responds to temperature. And of course, always thinking about what was the other thing I was gonna... Oh, just things like sweating, right? So you have the ability to put fluid onto the skin surface in order to dissipate heat, which again, not all animals do, right? Your dog doesn't do that. It has to pant. And so we actually have some pretty elaborate mechanisms for maintaining body temperature. Okay. That's it. Hopefully for most of us, this chapter is a review and not too hard, but this is some of, I think for a lot of folks, the more complicated materials that we come across. And so we will go through whatever we need to go through until this makes sense. All right. Hope you guys had a great weekend, and we'll see you soon.